thank you everyone for joining this session. I can see from the immediate polls that there is quite a spread of knowledge, so I'll try my best to try and cover as basic information of the technology all the way through to trying to show some real good applications and the true benefits for implementing this technology for a range of different applications. So the main areas I do want to focus on are sort of the benefits of the technology, and I'm going to cover it over a few different applications that should hopefully be of interest. So if I just to start off, what is pulsed electric fields? So just to try and be as basic and strip it back down to the bare essentials, it is a process of electroporation. So basically making holes in cells, and you can utilize it using low energy to punch these holes in cells, and they can be reversible or irreversible depending on the intensity of the energy that you use, but it does offer a broad range of applications for the food and drink sector. So the basic mechanism of how pulsed electric fields works is most cells, living tissue cells, will set up a natural ion gradient where they're controlling the nutrients and the ions and solutes that they want to get in and out of the cells for their own metabolism. As soon as you start to apply a high voltage electric field with those cells in between, you can then start to disrupt all the ions and you can cause polarization. As soon as you start to polarize those ions, you can then lead to pore formation within the actual membrane tissue of the plant cells or microbiological cells, depending on the application that you're trying to apply the technology for. And these pores can be irreversible, so you can completely break the cells open if you're looking for extracting compounds, or they can be reversible if you want to start to stimulate other different biochemical responses. So you can start to actually tailor the process depending on the application. So you can start to target the treatment. And here is just a list of some of the applications that you can really target this particular technology for. So in the lowest energy end, so 0.1 kilojoules per kilogram, you're looking at a stress response. So can you just deliver enough energy to stress some cells to change their metabolism or increase their metabolism of certain components that you then want to go on to extract or utilize more of? And there's some applications there as well with increasing fermentation, so being able to stress certain yeast or other bacteria in the culture, where you can then get them to actively grow a bit faster in the culture. Some good applications in fermenting ethanol and things like that, where you want a very fast fermentation. If we start to increase the energy, so we go up to 1 kilojoule per kilogram, you're starting to look at cell electroporation. So now we're starting to open up the pores and the tissues of the structure of the products that you can treat. So solid fruits, different root vegetables can have some really good applications for improving of cutting or opening the structure for drying applications, whether it's a hot air drying or freeze drying. And I'll talk in some more detail with some good examples of those as I go through the presentation. If you increase the energy further, then you're looking at cell permeabilization. So really opening up those structures, trying to improve the extraction. So you can see 2 bottles, one of them's filled to the top with apple juice. So you can start to extract more juice from less actual raw ingredients. Or you can start to extract more of the active compounds that you want. So things like when you're developing certain liqueurs or in wine, you can start to extract more of the important antioxidants or other compounds that are relevant for those types of products. For meat processing, there's applications where you can improve brining, meat texturization, so extracting some enzyme components that help to break down the meat and keep it more tender. And then I suppose from the poll that was launched by Richard at the start, a lot of you guys are interested in liquid processing, so cell inactivation of liquids, juices, smoothies and dairy, and you're going to much higher energies here for cell inactivation. And that's primarily because the organisms and the cells that you're trying to punch the holes in to inactivate them are a lot smaller, so we need to deliver the right levels of energy to make sure we can start to compromise those membranes. And then even higher energies, you can start to completely disintegrate cells, form sludge. So the first area I want to talk about is plant tissue processing. So I've only got a few slides on each, and I'll talk about a lot of the benefits that start to come with these applications and technologies. I'm a very visual person, so there'll be a lot of images. One thing I want to say from the start is we are working heavily and in partnership with food physics. So a lot of the images and the some of the graphics that have been shown in this presentation have been provided by food physics and Elia. So we're very grateful to their support and access to the technology that we've got for this year. So for plant-based processing of tissues, you can start to see here for a variety of different types of products. The most common one that's very well publicized now is sort of the effects on potatoes and being able to reduce the turgor pressure in the cells, because once you start opening the cell structure, you can let out certain liquids, but you can retain some more of the solid components, some more of the solids stay in the cells. But what it means is the product becomes more malleable, so you can start to bend it, you get less breakages, you get more reliable cuts, you get less feathering, and there's some even further downstream benefits of the technology where sometimes you might not even need a preheater, so you can get energy saving benefits as well as water saving. And with some of these products, when you're frying them, you're actually using the oil uptake is reduced, so you're using less oil. So you can be more sustainable in your use because you're actually getting more product through the door, so you can increase your yields as well as using less oil in the process, whilst making energy savings. Because I mentioned that you can actually punch holes and reduce some of the liquid, what you tend to find as well with applying pulse electric fields with some of these products is you can actually retain more starch in the products. So there's less starch in the water, so you're putting less pressure on your water treatment systems to try and clean that water back up if you're using it for reuse or discharging. So there's benefits for the process from that side of it as well. And obviously you can get some really great colour on the products. So because you're having less oil uptake, and you're not you're reducing the amount of starches that's coming out of the product, you can actually also start to see significant acrylamide reductions for a range of products. So here we've got carrot chips by about 19% reduction, all the way up to beetroot, which is about 50%. So you get much more of a brighter coloured product. So it's more attractive and more appealing, but also it's got less of the acrylamide compounds in it. So some real great benefits when you start processing plant-based tissues and then you use them for downstream processes. Another area that's widely used for pulse electric fields is extraction. So being able to punch holes in cells and open those cells up means you can start to extract more nutrients and more components out of the cells. So here's just an example of a phytocyanin compound. So it's supposed to have many health benefits when you can extract it from spirulina. And by applying pulse electric fields, not only can you increase that content, you can also increase the percentage purity of the content as well. So you're getting much more of that compound out, and you're being much more efficient with the cultures that you're using for the outputs that you're requiring. So some great applications for extraction with the technology. Here's an example where the technology is commonly used for sugar beet extraction. So applying 1 kV per centimeter squared, and then you're pressing at 5 bar, you can get a 70% increase compared to an untreated sugar beet. So it really does open up those structures and allows manufacturers to extract the maximum yields from their crops. So there could be great potential as well for waste. So if you've got some waste products where there's other active nutrients that are present in the products, you could potentially be extracting those and repurposing those for other value chains or making higher value products out of your waste if you can extract it. Here again shows some more examples of sort of extraction. So here's like a berry liqueur where you can see for the application of pulse electric fields, if you just look at day one, the left-hand side images. So the top one is for 100 grams of berries per litre compared to the bottom one, which is 300 grams per litre. You can start to see straight away that the pulse electric field can dramatically extract a lot more content and really increase that colour intensity quite dramatically. And what that means is for some of these applications is you can reduce that maceration time. so you don't need to leave it steeping in the product for as long to get some of those active compounds that you really want present in the product. And you can also potentially get the same sort of extracted compound for actually less berries. So because you've been able to extract more out of the berries themselves, you can actually reduce the concentration of the berries that you're extracting from to still deliver a similar product. So again, you could use more, you could make more with less and be more sustainable. So that's extraction. The next section I want to sort of highlight and talk about is drying benefits for the technology. So whether you're heat drying or whether you're freeze drying. And these just show a picture of some applications where pulse electric field's been applied and then freeze drying has occurred. And you can see the actual protection compared to an untreated product where you can really protect the structure. So there's some fantastic benefits with drying applications. So you can actually reduce the drying time. So here they're combining pulse electric fields and then a microwave drying of carrots, and that microwave drying time is reduced by 40%, so less thermal load on the product, less energy consumption. And you can see on the right hand side the pore structure is really opened up from the pulse electric fields, and that just allows that mass transfer and allows the ability for that to be removed from the product to allow it to dry quicker. And you can get better colour retention because you're not exposing it to excessive thermal loading. The benefits of the technology also can be applied to freeze drying. So different products may have different benefits. So this just gives you an example of a few different products where the drying time has reduced by applying pulse electric fields first. And the beauty of the way that the technology works is it's opening up that pore structure. So it enhances that water diffusion and then allows the structure to be maintained. And I'll show some examples of the structure of some strawberries in the next couple of slides. But as well as maintaining a higher quality, you're actually improving the process as well, reducing that drying time. So here's just some examples of freeze-dried products, just so you can get a feel for how pulse electric field can start to perform. And you can see that the structure looks much more like a fresh product. And that's primarily because it's opened up those pores more. It's not compromising the cell integrity as the product has been frozen down and dried off. And on this slide here, you can see an X-ray CT scan where the pore structure is much more open. So it allows that water to diffuse out and you can maintain that structure where you're not getting that product shrinkage during the process. So there's some really great benefits when you start to apply pulse electric field before you're then drying down products, whether it's freeze drying or hot air drying or drying with heat. So the last sort of area I wanted to talk around was beverage processing. I didn't put many slides on this area because I thought it was more well known, but it seems like a lot of you have much more active interest in beverage processing. So here's a system from Elia that does, I think, up to about 10,000 litres an hour, depending on the configuration of the system. And by applying high voltage pulse electric fields, you can start to break down and inactivate the microorganisms with minimal effects on the beverage that you're actually processing. So the main slide that I really wanted to showcase with this technology, and it links into some other work that we've done previously at Camden, is when you start to look at very heat sensitive compounds, particularly sort of green products tend to have very heat sensitive compounds. Here you can see freshly squeezed, which has a very short shelf life, but it has a brilliant vibrant green colour. As soon as you start to thermally treat, you always go to that khaki green colour. It never survives very well when you start to apply a heat process. And there's many other natural compounds that people are used to formulate beverages where heat just doesn't agree with the actual ingredient, and then you'll get that loss of colour. Whereas with technologies like pulse electric fields, because it is a very short exposure to a rising temperature, much reduced thermal load on the product, and you're using the pulse electric the high field strengths to punch the holes in the cells, you can get a much better quality retention. So you're getting, here you can visibly see it's as good as the freshly squeezed after about 3 weeks. So excellent colour retention, and we showed it with some previous reports on the member funding projects where we added a natural blue colouring, and we could get fantastic colour retention, and it was comparable to control over the entire shelf life. So the way that the pasteurization process works, you typically tend to preheat the product very slightly before it goes into the pulse electric field system. So about 30, 35 degrees, 40 degrees max. And then as soon as it passes through the chamber in a very short time, it's exposed to the high field strengths and the high voltages, you can then start to inactivate the microorganisms. And there's a slight synergistic effect of that slightly elevating the temperature, which gives it a much better shelf life. So you can start to really extend the shelf life now with pulse electric fields. The last area I wanted to touch on was for the impacts on fermentation. So I was coming across some data that showed the impacts where you can start to treat leaves and you can start to improve the fermentation process. So on this slide, they've studied looking at pulse electric field treating the leaves, and then they've been drying the product out, and then on the left hand image on the bottom you can see an untreated PEF where it's quite a dilute colour. If you pulse electric field treat and then you dry the leaves down, you can start to see that there's much more compounds being extracted from those leaves. If you actually pulse electric field and allow the product to ferment and have it dried down, then you can start to see even more compounds can start to be extracted. So but by being able to open up that pore structure, you are releasing certain enzymes that can help with the oxidation process. And you can see on the middle image, there's two products that have been pulse electric field treated at different energies, and they've got a really black color to them because they've undergone a rapid oxidation process. In the right hand side image, you can see control leaves on the left and pulse electric field leaves on the right, and you can really see that by releasing those enzymes from the cell tissues, you can start to dramatically accelerate that color development and that oxidation process, so potentially speeding up that process and then being able to extract more of the valuable compounds that gives the importance for the tea quality. So that's sort of the list of the applications I just wanted to touch on today. There are a number of other applications, as I've started to allude to, but there's other things on improving peeling, other extraction applications on olive oil where you can get some dramatic improvements. So there are a wide range of applications for the technology by being able to manipulate the structure of some of the tissues. And as I mentioned at the start of the presentation, we are working in partnership with Food Physics this year. So this is the system that's been recently installed at Camden BRI. And we're looking at doing some work with this system on our member-funded research project. And this system is a dual-based system. So at the moment, through the little window, you can see there's two white discs. So that's the continuous liquid setup, where it applies the high voltages to a liquid setup. But we can interchange that out for the bulk treatment system. So we can put in bigger buckets where we can start to treat at lower field strengths and look at more tissue manipulation or functional modification of certain tissues. And this year on the member funded research project, New Technologies for Food and Drink Manufacturing, we are looking at trying to explore some new applications with pulse electric fields. So historically, it's mainly been inactivation of microorganisms in liquids, so using high field strengths in liquids. And we're looking at trying to use combinations of pulse electric fields, maybe with some other stresses for surface inactivation of microorganisms on bigger food products that typically wouldn't have gone through those electrode systems. So we're very much looking to explore different combinations of microbial inactivation if it's possible for a range of different food products. We haven't completely fixed on the food products yet, So we are very much open to your feedback or any of your comments where you think there's challenges or things that you'd like or the technology to potentially explore. So we're very open for receiving feedback to see if it's something that's we think is worth exploring on behalf of the membership. So with that being said, I thank you everyone for your attention, and I'll hand back over to Richard, who will potentially open up for questions.
Okay, brilliant. Thank you very much, Danny. That was a fantastic overview of pulsed electric field processing and some of the applications that are available. So I'm sure it will have generated some questions among the attendees today. If you do have a question you'd like to ask Danny, then think about it now and add it to the Q&A window, and we'll come back to some of those at the end. So just to round off the presentation, I'd like to go through a few slides telling you how you can find out more about the work we do at Camden BRI. Related to processing technology. So first of all, newsfeed emails. Many of you are probably already subscribed to these because that's how you found out about the webinar in the first place. But if not, this is our email service for raising awareness of all of the work that we do. If you go to the link at the bottom of the page there, you'll have a whole range of options that you can select. And processing and preservation is probably the most relevant for this particular group. In terms of upcoming events, we have several conferences and seminars and training courses coming up before the end of the year, most of which are online at the moment. So whether they're related to microbiology or processing or safety and quality culture, we have an event for all of your interests. Similarly with training courses, if you're involved in thermal processing at all, several courses coming up before the end of the year. So all of the information there is available on the Camden BRI website. Some of you may be involved in our member interest groups. So if you're a member company, you're eligible to join these groups. They're essentially networking events for member companies. Previously, they would have been face-to-face, but they currently meet online. A whole range of different topics there. One based on processing operations and preservation, which could be of interest to you. That group might be worth exploring. The MIGs are actually running at the moment. The first one started yesterday, so most of them have still got a date coming up. So if you're interested in networking online with other like-minded member companies, then they're well worth joining. If you use social media, then we're active on all of the major platforms. Those are the links to the Camden BRI pages. That's all I'll say about that. So we're now at the end of the presentation, so we can come back to some of your questions. As I say, use the Q&A window if you'd like to ask something. We've got some that have come in already, so I'll bring Danny back in at this point. So the first question we've been asked, Danny, is whether pulsed electric field processing would be suitable for shelf life extension of soups. So they've mentioned smooth and particulated soups as specific examples. Are we able to say anything about that?
Yeah, we can touch on it a little bit. As far as soups go, it does depend on the type of organisms that you're trying to control, and also the type of particulates. So if they are large particulates, you normally need quite narrow electrode spacing. So you're just going to block the electrodes. So most of the products tend to be very smooth products that go through these processes. So you can think like smoothies, they tend to be quite smooth, that you don't tend to have many particulates. But if you're trying to target spores, for example, you're less likely to get a combination with spores unless you're starting to combine the process with higher heat. And then again, I think I'd have to go to the manufacturer with regards to sort of the levels of spore kill you might get if you did start to try and combine it with heat, because you might have to go to quite elevated temperatures. But for most of the vegetative pathogens, you can start to get decent log reductions of those with a smooth product.
Okay, excellent. So I guess to a certain extent, it's a case of judging whether or not it be suitable and then testing it to see what happens. Is that right?
Yeah, and I mean, what I would say is, I mean, for the new techs project that we're looking at for this year, we're trying to see if we can get microbial kill on certain surfaces of products in bigger environments. So there might be the potential for treating products that have bigger particulates, but I think that is something that is being more looked at and more developed. It's not as well established probably as the juices at this stage, but it's something that's definitely on the cards for being looked at.
Okay, excellent. So the next question we've got is about the effectiveness of the technology on targeting certain organisms. So would it inactivate yeasts and molds, or does it just target bacteria?
Yeah, so I probably should have mentioned it in the presentation, but yeah, so it's very good at the pathogenic species, but also the yeast and molds are affected as well. So you can get a significant delay in any sort of potential regrowth, which is why you get a really good extension in the shelf life. So it does affect yeast and molds as well as vegetative pathogens.
Okay, brilliant. So this next question's quite product specific as well. It's about drying fruit and vegetables, and whether or not it can provide any microbial pathogen lethality prior to drying. Can we say anything about that?
That's a very good question. One I do not have the answer to be honest. At the moment, I think the field strength there is mainly just to open up the pore structure without damaging it. So it might not be strong enough field strength to cause microbial inactivation. But it is something we're trying to look at with regards to how we're going to combine it to get surface inactivation. And could we even move to bulk food products like that, or are we going to start to open up the pore structures too much and cause too much quality issues? So it's something we're looking at exploring with the manufacturers at the moment to see where the applications might be if we're trying to do surface inactivation. But at the moment, I don't think there's any microbial control at that stage.
Okay, thank you. We've got a question about the relative speed of the processing sort of technology. This one comes from a company that processes liquid eggs, and they currently use pasteurizers to do their processing. They're interested to know the speed that they can typically process at. They're currently using 6,000 litres an hour. Can pulsed electric field processing do anything comparable?
Yeah, I mean the systems that the big system that I showed when I started to talk about the beverages, they can be adapted up to 10,000 liters an hour. Whether the specific things you have to do with regards to eggs to get them to process, I'd have to refer back to the equipment manufacturer on that. But as with regards to the flow rates, they're getting up to 10,000 liters an hour now for juice applications. So they're within the right window of operation for that type of flow rate.
Okay, thank you. So we've got a question here asking about whether or not a pilot scale machine is available. Presumably that means that at Camden BRI, is the equipment available for pilot scale trials?
Yes, so we have it for the member funded research, but while we have the system on site for the member funded research, we offer it out for any trials that companies want to try and undertake on it. So it gives people a way of accessing the technology without having to rent it or have the hassle of having it on their own site. We can start to help them perform some trials or independent trials to help them generate some independent data.
Okay, excellent. So we've got another product specific question here, this one about crisps. It's asking if pulsed electric field processing can help with acrylamide or oil pickup in crisps. Have we got any experience of that?
We've done some stuff previously a few years ago looking at the impacts on PEF processing on sort of fries rather than actual crisps, so it's more small chips. But the data that I've seen from some of the manufacturers and some of the data I've come across, there is definitely a potential for being able to reduce the oil uptake in some of those products. It does depend on other parts of the processes, what's going on, but you can get some reduced oil for those types of products. And the other question, part of that question, what was it?
It's about acrylamide.
Acrylamide, yeah. So what we did find with the chips and the frying, if we standardized everything with regards to chip size and everything else, you definitely do see with pulse electric field a lightening of the product. So although we didn't specifically test acrylamide when we had the system on testing, as you can see for the pictures that I shown before for other products with sweet potato and everything else, you can get that reduced acrylamide for the same sort of frying operation.
Okay, thank you. So we've got a slightly more general question now. It's asking if you need to have high moisture content to be able to use pulsed electric field processing. I'm guessing it helps, but are you able to comment?
That's a really good question. I think the old viewpoint would have been yes. I think some of the conversations I've been having doesn't, or it can be quite nuanced. We've seen some applications and discussed about some applications where you can potentially treat drier materials and you can still elicit holes in the outer texture, so you can improve the rehydration of those types of products, so you can get a faster rehydration by processing with pulse electric fields. So although it's not penetrating completely into the product, it might start to be affecting the outer structure where you can still penetrate or improve the penetration into the product. So there is the potential for dry products to have an impact with pulsed electric fields, but it does depend on the product and the way that you're applying the technology.
Okay, thank you. So we've got a question now about academic collaboration. I'm guessing this has come from a sort of an academic institution. They'd like to know if there's any potential for collaboration on this type of work. It might be a conversation to have at a later date, but is there anything we can say at this stage?
I'm always open for collaborations and understanding how to work with other people or leveraging the work that we're doing for the benefits of our member companies. So I'm happy to talk about other ways of collaborating and combining expertise to try and maximize results and trials. So yeah, happy to have a chat offline if the person wants to follow up with me direct.
Okay, excellent. And we'll make this the last question because I'm conscious that that time's pressing on. So it's once again about collaboration. So we've mentioned the member-funded project as the initial starting point for this. Is there a way that sort of companies can get involved separately? So perhaps with one-to-one trials or something like that?
Yeah, so obviously we're very interested in trying to finalize the product so people can support us a little bit on the member funded research. But if you need to access a completely different area of the technology and you want to try some specific stuff on your products, then by all means get in touch and we can have a discussion around it and we can do private confidential trials. If there's a big group of challenges that's facing the industry, we're happy to set up club models where everyone chips in a little bit of funding, and then we can do a bigger piece of research for a consortium of companies where they share that knowledge within the consortium. Yeah, so we're open to different ways of working depending on what the challenges are. We're trying to set up a pulse electric field club for alcoholic beverage processing at the moment. So I'm working with one of my colleagues, Greg Jones, to try and see if we can get enough companies interested in that. So if you are interested in that space, then please as well get in touch because we're very interested in trying to set up different ways of collaborating to access the technology.
Brilliant. So there's clearly a lot of potential then. Okay, so that brings us to the end of the session for today. We've just been talking about getting in touch. If you'd like to contact either one of us, then you have our contact details in the presentation. So all that remains to be said is thank you very much for joining us today. Hopefully you all managed to get something out of the presentation and can see some potential applications for you and your company. So thanks for joining us. We'll hopefully see you again at some of our future webinars. So enjoy the rest of your day and all the best. Thank you.